For 70 years, the question everyone asked about the Dead Sea Scrolls was the same one. What do they say? Scholars pored over the handwriting, the language, the theology hidden inside thousands of fragments, trying to reconstruct the world that produced them. Almost nobody thought to ask a much simpler question. What are they actually made of? When a molecular biologist finally did ask that question, the answer didn't just add a footnote to the field. It quietly undid one of the most confidently held theories in biblical archaeology. A shepherd, a stone, and an accident of history. The story starts in 1947, when a young Bedouin shepherd named Muhammad ed-Dib was searching for a stray goat near the cliffs above the Dead Sea. Looking to scare the animal out of hiding, he tossed a rock into the mouth of a cave and heard something shatter inside. When he climbed in to investigate, he found tall clay jars, some intact, holding leather scrolls wrapped in linen that had sat undisturbed for nearly 2,000 years. He had no idea what he'd found. Neither, at first, did anyone else. Over the following decade, archaeologists combed 11 caves near the ruins of Qumran and recovered roughly 25,000 fragments, representing something close to 900 separate manuscripts. Among them were the oldest known copies of biblical books like Genesis, Isaiah, and Psalms, alongside community rulebooks, hymns, and apocalyptic visions describing a coming final judgment. Radiocarbon and paleographic dating placed the material across a window from roughly the 3rd century BCE to the 1st century CE, a period covering the most turbulent chapters in the history of both Second Temple Judaism and the earliest Christian movement. The scrolls had survived the destruction of the temple, the fall of Jerusalem, and centuries of empires rising and collapsing above them, sealed away in some of the driest caves on earth. The theory that held for 70 years. For most of the 20th century, the dominant explanation for the scrolls was straightforward and, on the surface, extremely persuasive. Ancient historians like Josephus and Pliny the Elder had described a Jewish sect called the Essenes, an ascetic, tightly organized community devoted to ritual purity and the expectation of imminent divine judgment. The ruins at Qumran, sitting just below the caves, looked very much like the kind of settlement those ancient writers had described. Sectarian texts among the scrolls described a self-governing religious community with its own legal code and its own solar calendar, deliberately at odds with the lunar calendar used by the priesthood in Jerusalem. Apocalyptic writings, like the so-called War Scroll, described a final cosmic battle between the sons of light and the sons of darkness, language that fit neatly with a breakaway group explaining its own exile from mainstream religious life. The pieces lined up well enough that the theory hardened into consensus. The Essenes lived at Qumran, copied their scripture there, and buried their collection in the surrounding caves when Rome crushed the Jewish revolt around 68 CE. That explanation was taught in seminaries, repeated in museum placards, and treated in documentary after documentary as settled fact. The cracks nobody could quite close. Underneath that tidy narrative, though uncomfortable details kept surfacing, the kind that traditional paleography and textual analysis simply couldn't resolve. The scrolls showed enormous internal variation, different handwriting styles, wildly different levels of scribal skill, and most puzzlingly, multiple competing versions of the very same biblical books sitting side by side in the same caves. The book of Jeremiah, for instance, exists among the scrolls in 2 substantially different forms, one closely resembling the standard Hebrew Bible familiar today, and a noticeably shorter version with entire passages missing, and its chapters rearranged. That kind of textual diversity sat awkwardly with the image of one small, disciplined, doctrinally rigid community. When radiocarbon dating was finally applied to a sample of the fragments in the 1990s, some of the scrolls turned out to predate the Qumran settlement itself, further straining a theory that depended on one community producing and storing everything in one place across one continuous span of time. A few scholars floated alternative explanations over the years, suggesting Qumran might have been a pottery workshop, a seasonal estate, or a shared depository where worn-out sacred texts from multiple communities were brought for respectful burial rather than continued use. None of those alternatives had the kind of hard physical evidence needed to unseat an entrenched consensus. That evidence, as it turned out, wasn't going to come from another historian or archaeologist. It came from a geneticist who wandered into the field almost by accident. A lunch conversation that started something new. In 2012, Tel Aviv University molecular biologist Oded Richavi, who spent his career studying inheritance in microscopic roundworms, found himself seated next to biblical scholar Noam Mizrahi during a new faculty orientation event. The conversation drifted toward the maddening puzzle of the scrolls. Thousands of disconnected fragments with no titles, no page numbers, and no reliable way to determine which pieces belong to the same original document beyond comparing handwriting by eye, a method that was useful, but inherently subjective and occasionally wrong in ways nobody could catch. At some point in that conversation, Richavi asked a question nobody in the field had seriously pursued. What exactly is the parchment made of? The answer was animal skin, and animal skin carries DNA. That single realization opened up a method nobody had thought to apply to sacred scripture before, borrowing techniques from ancient DNA research that had already reshaped the study of human migration and prehistoric population movement. Richavi teamed up with Mizrahi, later joined by geneticist Matthias Jacobson of Uppsala University, biomedical scientist Christopher Mason, and Nina Shore, who oversaw the scrolls as curator at the Israel Antiquities Authority. Together, they spent roughly 7 years chasing a question no one had ever tried to answer this way: reading genetics without touching the text. The obstacles were significant from the start. The scrolls are irreplaceable and sacred to multiple faiths, which ruled out any method involving cutting or drilling into the parchment itself. The workaround came from an unlikely source. Decades of careful handling had left fine parchment dust in the bottom of storage containers, dust that still held collagen, the structural protein in animal skin, with genetic material locked inside it. By collecting dust from blank margins and worn edges, places where no writing had ever existed, researchers could gather usable samples without touching a single letter of the actual text. Even then, the science was difficult. The parchment-making process itself, soaking, scraping, stretching, and drying animal hide, damages DNA at a molecular level, and the iron-based inks used on many scrolls are chemically corrosive. On top of that, the scrolls had already endured 2,000 years in desert caves, followed by 70 years of handling by researchers, each contact a potential source of modern contamination. The team worked in dedicated ancient DNA labs, essentially clean rooms with protective suits and UV sterilization, running samples through multiple independent extractions and cross-checking results between separate laboratories to rule out contamination before trusting any result as genuinely ancient. The cow that broke the theory. Most of the samples came back exactly as expected. Sheepskin, the standard parchment material across ancient Judea, taken from animals well suited to a harsh desert environment. But a handful of fragments, including 2 pieces from the book of Jeremiah, came back as cowhide, and that single detail turned out to be far more disruptive than it sounds. Qumran sits in one of the driest, harshest environments on the planet. Sheep and goats can survive there on minimal water and scrub vegetation. Cattle cannot. A single cow requires large amounts of water daily and real grazing pasture, resources the Judean desert around Qumran simply didn't have. That meant any scroll fragment written on cowhide had to have originated somewhere else entirely, somewhere with real grassland and water, and been physically transported into the Qumran caves rather than produced locally. According to Richavi, this single genetic detail effectively disproved an earlier assumption that one of those cowhide Jeremiah fragments belonged to the same scroll as a companion piece made of sheepskin. Genetically, they couldn't be part of the same document at all, and textually, researchers found the 2 Jeremiah fragments represented different versions of the prophetic book, not simply variant copies of one shared original. What the genetic fingerprints revealed. Beyond identifying species, the researchers could compare mitochondrial and nuclear DNA across samples to determine whether separate fragments came from closely related animals, potentially the same flock, which would suggest they were produced at the same place and time. Some fragments did cluster this way, pointing toward shared production centers. But other fragments showed no genetic relationship at all, coming from entirely different animals raised in entirely different places. Researchers also found that certain widely known texts, such as the liturgical composition known as the Songs of the Sabbath Sacrifice, existed as multiple copies made from genetically unrelated animals, suggesting the text had been popular enough to be copied independently in several different locations rather than guarded as the exclusive property of one isolated group. Put together, this pattern doesn't look like the output of a single scriptorium serving one disciplined community. It looks like a collection assembled gradually from multiple points of origin, representing multiple communities across a wide span of time that a shared library, not a private archive. If the scrolls really had come from one isolated sect at Qumran, the sheer diversity inside the collection is hard to explain. Why would a tightly controlled religious community bother preserving multiple competing versions of its own sacred texts, including versions reflecting theological positions it supposedly rejected? The genetic evidence lends support to an alternative explanation that a minority of scholars had floated for years without solid proof. That the caves functioned as a geniza, a sacred storage site, where worn-out or superseded texts bearing the name of God were respectfully retired rather than destroyed, in keeping with Jewish tradition. Jerusalem sits close enough to Qumran that Jews fleeing the city during Rome's siege between 68 and 70 CE could plausibly have carried sacred scrolls out into the desert. Adding them to material other communities had already been depositing there over generations. Under this reading, the caves weren't a private theological archive belonging to one breakaway sect. They were closer to a shared communal library reflecting the full, messy range of Jewish religious life in the final years of the Second Temple period, a period during which the earliest Christian movement was also taking shape. Why this actually matters. The implications go well beyond settling one archaeological argument. If multiple, sometimes contradictory versions of Jeremiah, Genesis, and other core texts were circulating simultaneously among different communities, then the idea of a single, fixed, universally agreed-upon scripture during this period doesn't hold up. Canonization wasn't a finished, top-down process. It was still actively contested, with different communities each treating their own version as authoritative. The genetic data gives that idea a physical foundation it never had before, since it can now show, rather than merely argue, that separate communities in separate places were maintaining separate scriptural traditions at the same time. That has real consequences for how historians understand the religious world into which Jesus was born and the earliest Christian movement first emerged. Not a settled, uniform tradition, but a landscape of overlapping, sometimes competing communities, each convinced it held the authentic version of a still evolving sacred text. Where the research goes from here. Published as a cover study in the journal Cell in 2020, this genetic work opened an entirely new avenue of research that scholars are still actively pursuing. Researchers are now expanding DNA analysis to a larger set of fragments, combining it with multispectral imaging capable of recovering faded or invisible writing, and building broader reference databases of ancient animal DNA from across the region. The long-term goal is ambitious. A genetic map precise enough to indicate roughly where in the ancient Near East a given fragment's parchment originated, and by extension, where and among whom a particular scroll was produced. A story written in skin. What started as a casual conversation between two researchers over lunch turned into a genuine correction to 70 years of scholarly consensus. Traditional tools, handwriting comparison, textual criticism, physical examination of the parchment, had taken the field remarkably far, but some questions were always going to be out of reach without a fundamentally different kind of evidence. Reading the genetic material embedded in the animal skins themselves finally supplied that evidence. The Dead Sea Scrolls, it now appears, were never the exclusive property of one isolated desert sect hiding from Rome. They represent the accumulated, contested, and genuinely diverse sacred literature of ancient Israel, gathered piece by piece from multiple communities over a span of decades or longer, and finally sealed away as the world that produced them came apart. A shepherd's stone opened the door to that world in 1947. It took a molecular biologist, a chance conversation, and 7 years of careful lab work to finally hear what was waiting quietly behind it.